Getting rid of cancer might eventually mean getting a couple of injections of your own engineered cells. By using CRISPR technique, researchers expect that more than 3,000 genetic disorders, including cancers, are gonna be cured. The word CRISPR is an abbreviation for clustered, regularly interspaced, short, palindromic repeats. CRISPR is a gene editing tool that can cut DNA precisely, turning genes off, adding new genes, or changing their functions. The CRISPR method is based on the natural system used by bacteria to protect themselves from infections by viruses. When the bacterium detects the presence of virus DNA, it produces two types of RNA. One of them contains a sequence that matches the invading virus DNA. These two RNAs form a complex with a protein called Cas9 that works as an enzyme to cut the virus DNA. When the guide RNA finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying the system realized that it could be engineered to cut any DNA sequence at a precisely chosen location by changing the guide RNA to match the target gene. And this can be done not only in a test tube, but also in the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock into a short sequence known as the PAM. The Cas9 will unwind the DNA and match it to its target RNA. When the match is complete, the Cas9 will use two tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut, but the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise. For example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. That mechanism gives us the means to edit your immune cells and make them better cancer hunters. Cancer occurs when cells refuse to die or keep multiplying while concealing themselves from immune system. To treat cancer, CRISPR is used to remove two genes from the patient's T cells, which are immune cells. One of the genes to be removed makes a checkpoint called PD-1 that cancer cells use to put brakes on the immune system. The other edit deletes the receptor that immune cells normally use to sense danger, like germs and sick tissue. An engineered receptor added in its place will instead steer T cells toward particular tumors. These edits allow the immune cells to more easily attack cancer, inhibiting the brakes and allowing the cells to fight off the cancer. Once the edits are made in the lab, the patient's cells are infused back into their body. Things are picking up pace quickly. In a decade or two, we could possibly cure thousands of diseases forever. With a powerful tool like CRISPR, we might be able to end more than 3,000 genetic disorders. Researchers are already building a modified version of Cas9 that's made to fix the genes that cause diseases. However, CRISPR can and probably will be used for much more. The creation of modified humans, for example, by infusing CRISPR into reproductive cells or very early embryos. Designer babies mean gradual but irreversible changes to the human genome. The thing that will bring many ethical questions and ethical issues to the biomedical community.